Okay, so we're going to look at the process of aerobic respiration. So let's start with what we already know from GCSE. So we know from GCSE that in aerobic respiration, glucose reacts with oxygen to give us carbon dioxide, water, and energy. And we should also know, or you should also know, that this can be written as C6H12O6 plus 6 oxygens gives us 6 CO2s plus 6 waters plus energy, which we now know from A level is actually the molecule ATP, adenosine triphosphate. Okay, if you don't remember what adenosine triphosphate is from the beginning of the course, remember that it is made up of a ribose sugar attached to a base with 3 phosphate groups associated with it. It is what we call a nucleotide derivative because it is very similar to the nucleotide structure that we find within nucleic acids. And it is the breaking apart of the phosphate bond that will provide energy for specific reactions. So what we now need to think about is how this process actually takes place, because it's obviously more complicated than shown here. So first of all, let's just think about the structure of a cell. So we know that within a cell, okay, we've got obviously our nucleus in the middle. I'm not going to draw all of the organelles, but we know that we have these mitochondria, which we always talk about as being the site of respiration. Well, actually, that's not entirely true. It's the site of aerobic respiration. And that's because aerobic respiration actually takes place in 3 parts. Part number 1 is glycolysis, which takes place in the cytoplasm of the cell. Nothing to do with the mitochondria at all. Part 2 is the link and the Krebs, which takes place in the matrix of the mitochondria. And part 3 is oxidative phosphorylation, which takes place on the cristae, those inner foldings of the mitochondria. You should have come across the structure of the mitochondria way back in cells, but we will have another look at it again when we get to that reaction. So the first stage that we're going to look at is glycolysis. Now, glycolysis, as we said, takes place in the cytoplasm. And in fact, when you watch the anaerobic respiration video, you will see that it is a process that occurs both in anaerobic and aerobic respiration. So we're going to start with our hexose sugar. We've just called it a hexose sugar. Obviously, that could be glucose, but it could be one of your other hexose sugars as well. That hexose sugar is going to be phosphorylated, and it's going to be phosphorylated into a hexose biphosphate, which means it's going to have 2 phosphate groups added. Now, where do we get those phosphate groups from? We're going to get them from ATP. So we're first of all seeing that ATP will get hydrolyzed into ADP and PI, and that phosphate will be added to the hexose to make a hexose biphosphate. And one of the first things you learn about ATP is its ability to phosphorylate molecules to make them more reactive. So what will then happen to hexose biphosphate? Well, hexose biphosphate, which is actually a 6-carbon compound, will then break down into 2 lots of triose phosphate. Triose, obviously a 3-carbon compound. So we've now got 2 3 carbons. So we're still with the same number of carbons as we had before. Now that triose phosphate will then be oxidized into 2 lots of pyruvate, which is also a 3-carbon compound. Now, because it's being oxidized, that is because it is losing hydrogen ions. And those hydrogen ions are going to be picked up by a coenzyme called NAD. So it will pick up those hydrogen ions and turn into reduced NAD, or NADH plus H+. Also in the process, you release energy, and that energy will be used to join 4 ADPs and PIs to make 4 ATPs. So if we have a look overall, in the overall process, we started off with our hexose sugar. And by the end of glycolysis, we've made 2 pyruvates. We've made 2 reduced NADs. And we've also made 2 ATPs. Now, why have we made 2 ATPs? Well, let's go back and look. We've had to put in 2 at the beginning, and we got 4 out at the end. So that is a net gain of just 2. So glycolysis itself is not very efficient in creating ATPs, in harnessing energy. And that is why, as we'll see when we look at anaerobic respiration does not actually cause the production of many ATPs, only 2. In order to produce a lot of ATPs, you're going to need to do aerobic respiration, because you're going to have to go into what we call oxidative phosphorylation. So this is a good point at which to start to look at the structure of our mitochondria, because we're going to need it for the next 2 stages. So if we remind ourselves of what a mitochondrion looks like and we can label it, that would be very helpful. So we have our outer membrane. And then our folded inner membrane. And when it's folded, we call them cristae. The inside is called the matrix. And then there are some other crucial things that are useful to know. So first of all, you will have ribosomes present. We believe that mitochondria were originally organisms that existed on their own, a form of prokaryote, and therefore it has their own ribosomes, and circular DNA can do its own protein synthesis. You will also see poking out of the cristae things which we call stalked particles, which we'll come across in oxidative phosphorylation. So we now know that the pyruvate and the reduced NADs, they are going to have to enter the mitochondria, because they're going to be needed in the next stages, the link and then oxidative phosphorylation. So let's go on to the link. So the link is called the link reaction because it links glycolysis to Krebs. So link starts with pyruvate. Now remember, pyruvate entered the mitochondria from the cytoplasm. The mitochondria can take in pyruvate. It cannot take in glucose, because glucose is too large. And that's a very common exam question, is why can it not take it in? In addition, it wouldn't be any use because glucose cannot be broken down in the mitochondria. It's only broken down in glycolysis, which takes place in the cytoplasm. So we have our pyruvate. Pyruvate, as we said before, was a 3-carbon compound. Pyruvate is going to form, and we've got 2 of them, is going to form acetate. Acetate is a 2-carbon compound, and therefore you must release those extra carbons as carbon dioxide, 1 for each molecule of pyruvate. In addition to this, this process involves the loss also of hydrogens, and therefore you will produce more reduced NADs. Now, I'm not going to worry about the exact quantities of reduced NADs. It's not crucially important for what we're doing. It's just the principle that we are producing reduced NADs. That acetate will then go on to join with another molecule called coenzyme A, and together they will make acetyl coenzyme A, again 2 of them, which will be ready for the Krebs cycle. Now, when we look at the Krebs cycle, I'm going to follow one acetyl coenzyme A through the cycle. So we know that the Krebs cycle takes place, and as does the link, within the matrix of the mitochondria. Now, it is a very, very complicated process, but at AQA A level, you do not need to know it in detail. Okay? And you do not need to know all of the different substances that are involved in the Krebs cycle, but feel free to go and have a look. There are many intermediates. We're going to start here with our acetyl coenzyme A. And as I said to you, we're going to only follow one through. We're also not going to be that bothered about the numbers of the different things we're producing. As I said, it is an idea to understand it rather than worry about the numbers. So acetyl coenzyme A will join with a 4, you write that a little bit more clearly, will join with a 4 carbon compound to form a 6 carbon compound. You do not need to know their names, but feel free to go and look them up. That will get converted into a 5 carbon compound, which will then get converted into a 4. During this process, we are going to be losing a carbon, so therefore we are also producing CO2s. And during this process, we are also going to lose some hydrogens. So we will produce reduced NADs, as we did before. We will also produce a compound called reduced FAD, and 1 solitary molecule of ATP per round of acetyl coenzyme A. The main idea of the Krebs cycle is that we want to make the reduced NADs and the reduced FADs, okay? So that is why we do the Krebs. Because we're going to need those reduced FADs and reduced NADs for the third part, which is oxidative phosphorylation. And that probably is the most complicated part of the process. Now, why the first step to ask us is, why is it called oxidative phosphorylation? You are going to oxidize hydrogen to form water, and you are going to phosphorylate ADP to form ATP. Now that leads us to something quite interesting to just discuss, which is we're going to make a lot of ATP in this oxidative phosphorylation. But if we scroll back up here, we saw that an ATP was made here and also in glycolysis. These types of ATP is made in what we call substrate level phosphorylation. In other words, the conversion of one compound to another is releasing energy to cause the phosphorylation of ADP. Quite a different process to the process that we're going to see over here. So this takes place, if you remember, on the Christie. So let me just put it in context. We're on the Christie over here. I'm going to take just a cut through of that membrane. So if we draw it out, we've got a normal membrane structure. So we've got our heads and our tails, our hydrophilic heads and our hydrophobic tails, like there. But then interspersed in this membrane are some electron carriers. Okay, so I'm going to put a few in. I'm going to draw some more phospholipids in so that we've got an idea of where we're sitting, like so. And then the last molecule in this chain, we'll make it a different color, is this molecule over here, which is called ATP synthase. And that's what made our stalked particles that sort of stuck out like lollipops. So if we put where we are, this is obviously the matrix over here. And down here is the intermembrane space. So what's going to happen here is actually reasonably simple. The reduced NADs or the reduced FADs are going to come along and they are going to be oxidized. And they're going to be oxidized by releasing their electrons to the first electron carrier and releasing their hydrogen ions in the middle. Okay, so that is the first stage that's going to take place. The electrons will then be passed down the series of electron carriers by a series of oxidation and reduction. And during that process, the electrons will lose energy. And as they lose energy, that energy will be used to pump protons through these electron carriers. That builds up the concentration of protons in the intermembrane space. So they will then diffuse back through ATP synthase. And as they diffuse back, the energy that they give off will be harnessed in the form of ATP. The hydrogen ions and the electrons are then picked up by oxygen, which acts as our final electron acceptor and forms water. So if we write that so that it's sort of easy to understand, we start off by saying reduced NAD and FAD are oxidized and release their protons and electrons. The electrons are passed along a series of carriers and lose energy. Energy is used to pump protons through the membrane into the intermembrane space. The protons then diffuse back through the ATP synthase, causing the production of ATP from ADP and PI. Never forget to say where it's coming from. Oxygen then acts as the final electron acceptor and joins with the protons and the electrons to form water. Okay, it is worth knowing, because it's mentioned in the specification, that the protons going back and forth is called chemiosmosis. The osmosis is the movement of the protons through the membrane and their diffusion back again. This idea about it acting as the final electron acceptor is really important. It comes up all of the time. All right, it's given a mark every single time. If we don't have any oxygen, then we don't have our final electron acceptor. So if this will link to the next video, no oxygen equals no final electron acceptor. That means the whole of oxidative phosphorylation grinds to a halt. And therefore the whole of Krebs grinds to a halt because you've got no need to make reduced NAD and reduced FAD, which means the only thing you'll do is glycolysis, and you're going to hit anaerobic respiration instead. Okay, so I hope that is you have a good understanding of the process of aerobic respiration from that.